So, good afternoon. Thank you to each of you for being with us today. I'm Loïc Chabanier from UI, and I will be serving as your moderator for this session. So, what is the topic today? The topic of this roundtable is to discuss the current state and future of AI in drug discovery, with a special focus on digital twin. And the question is, are digital twins a hype of the day or a healthcare revolution? So we will discuss this with the panel and try to answer to these, I would say, basic questions. So let me introduce the panel, starting with David. David, you are the former Chief Solution Officer at Hopkins and the co-founder and general manager of a startup called Bioptimus, based in France. And you are working on developing a universal foundation model for biology to accelerate biomedical R&D from diagnostic from discovery to diagnostic. Thomas, you are the Chief AI Officer of Lilly, based in the US, 150 years old medicine company, the maker of popular cardiometabolic drugs and the first pharmaceutical company that hit 1 trillion milestone in value. Even in dollar, it's amazing. And Lilly is investing in supercomputing to train AI foundation models that will help in designing novel molecules as well as developing and delivering new medicines. And finally, Szabi Nagy from Budapest, Hungary. You are the co-founder and CEO of Turbine. And Turbine is developing a cell twin to simulate how cancer behaves and design effective therapies and therapy combinations. And I want to apologize for Charlotte. Charlotte could not be with us today. She got sick, the flu, and she apologized for not being here. And Charlotte is a professor in computer science and life science at EPFL in Zurich. So let's start, and I will start with you, Thomas. To prepare the next generation of drug, Lilly announced a huge investment in infrastructure and technology like digital twins and agents. So let's focus first on digital twins. And my first question to you will be, what are we talking about? In a few words, can you clarify the concept and describe the main future of this tool and what it brings in terms of drug developments?
Of course. So first of all, thank you for the invitation. Thank you everyone for coming. It's really great to be here in Europe. As you hear from my accent, I'm from Austria, so I'm always very fond of actually pushing forward Europe in terms of AI. So at Lilly, as you said, we built the world's most powerful supercomputer to actually drive all kinds of AI developments. And what is especially interesting about digital twins is that it's not chatbots. A lot of people, of course, think AI is only chatbots, but there's, of course, a much, much larger world of AI outside of chatbots. So in our case, when we think of digital twins, these are digital replica of physical, biological processes at large. So that could be, in the extremum, a patient, could be a cell, could be a manufacturing process, or could be a simple statistical model. So it really always depends about the granularity when you talk about digital twin. You can model things on a very low granularity with a few parameters, or something to a very extreme granularity. And with a cell or a human, of course, we are far from having perfect modeling. So at Lilly, we do that in the cell space to try to predict better properties of models we design. But we do that also a lot in manufacturing, where we actually have digital twins, not only of machines and manufacturing lines, but also of manufacturing processes. For example, we had a digital twin to improve the drying process of medicines, these APIs, and that led to millions of doses more. We could actually deliver to millions of patients more. So digital twins and AI, for example, in manufacturing, in the pharmaceutical industries, delivers enormous ROI today. While of course in discovery, which is very, very interesting for me as a scientist, of course, there's always much more time and we'll have biology play out that will take a few years. But in all these areas, of course, digital twins are important, and the new models we have, these very large deep learning models that allow us to train for much, much more data, these higher capacity models allow us to do much more fine-grained modeling of these digital twin models.
And I think what is very interesting is that you put it in place and it's at scales. And what do you see as the main challenging to make it happen long term?
So the first challenge was of course compute. We solved that now, so we can train at very large scale. Then the second one, when you actually train these models, is always, like in big organization, organizational change management. We have to have all the chemists and the line engineers actually use these models in the long run. But I'm very, very optimistic that we can actually expand in all these areas. A limiting factor in our industry is still always data. We are at a very luxurious situation at Lilly because we have decades and decades of data, also of failed experiments. So for example, if you just train your AI scientist on all publications out there, you will only have positive experiments. And that's a very tiny world, of course. So if you can actually train from all your failures, then you can build much better twins. That said, we of course still have much less data in medicine making than you would have, for example, in language out there. So being able to build automated labs to actually produce data, real world data, much faster than to train your digital twins much better, that you can propose new molecules and new approaches, that's the biggest bottleneck, and that will lead to that virtuous loop.
Thank you, Thomas, and I will come back to you later. Sabi, Turbine has established itself as a key player. You have signed partnership with top 20 pharma companies and successfully completed several major fundraising rounds. Can you give us a high-level overview of the main activity you are following, but also the ongoing research programs?
Yeah, absolutely. So, hi everybody. Great to be here, as always. So what we build at Turbine is essentially a virtual cell. We actually called it a simulated cell when we started, but we were a couple years before everybody else thought this was actually going to be a feasible idea, and now we've swapped to the more fashionable choice. We're just going with the times. But the core idea behind Turbine is to essentially build something that can predict the behavior of cells that you would test on in a laboratory setting, and also hopefully learn something so fundamental about the underpinning, the mechanisms of the biology of these cells that you can actually go beyond what's possible in the lab, and you can create more complex models, representations of 3D structures like organoids, or even animal models, and one day hopefully sort of tie into the world that David will talk about when it comes to predicting patient behavior. And so in a way, we're going bottom up and trying to understand the molecular underpinnings of cells, so we can actually train a model that predicts the outcome of experiments that you've never run before. And it's something that we spent a lot of time on over the past decade, essentially since we spun out of university. But one of the things that we've really learned about in the past year, and this is a big realization that I wish we had sooner in a way, is that it's amazing to talk about a virtual cell and virtualizing cells, and more people are saying that this is a great idea. But the biggest challenge is, A, of course, how do you train such a model that makes sense? And B, even more importantly, what good is it, right? What's the use of such a model? And for the longest time, I think actually we as a company, at least we as a team, have struggled more with the second part. So we've been able to create models that were able to model the behavior of more and more types of cells, more and more types of laboratory models across more and more disease areas over the years. But what really eluded us was actually how to make this useful truly, and how to find a way to actually make our colleagues in pharma see the benefits beyond just an interesting tool that can give you a few ideas that you may not have had, so as a source of novelty. And it's something that this year has really transformed the way we interact with pharma, actually, where we realized that actually the best way to start with such a new idea that you're running virtual experiments by the 100 millions is to dress it up in a very familiar way and say, listen, you're essentially we can virtualize assays that you have in a lab today. We can virtualize the experiments you run in the lab today. And first up, don't believe us when we say we can predict something in a patient or in a more complex model. Just think about whether you can have this as a way to make your experimentation more rational. Essentially, turn an empirical process of discovery based on literature and what you see in a few experiments here and there that you can actually run in a feasible drug discovery format, and turn into something that's much more engineering-like and much more rational by essentially simulating every potential experiment you could hope to run and finding the most promising ones and running those. And this integration of simulations with the actual laboratory workflows is something that we're working on mostly today, and this is what has driven sort of AstraZeneca's adoption of our technology in the ADC world, which is of course a very difficult sort of engineering challenge where there's too many experiments to run feasibly, even in a laboratory setting. And also, I'll talk a bit later about our work with Merck, which hopes to prove that we can actually also go beyond laboratory models.
And I assume that data is critical. So in terms of data collection, what is the strategy?
Yeah, no, good question. So in terms of the bottlenecks that Thomas also mentioned, data is clearly, I think all of us will agree, data is a key limitation. We, I think, have a bit of a tweak on this view where we think that, if you want to create technology that's useful today, you kind of have to accept the limitations of the data that you actually have access to today. But that doesn't mean that you just say, okay, I'm building a bad predictor. But for us, it has meant that you put in the enormous amount of unsexy work of harmonizing data sets that that have been generated in a multitude of different assays from different sources in different perturbation types, and try to essentially train a single model on this very icky, yucky, dirty, and sort of large set of biased small data sets, essentially. And that's something that we've spent a ton of time on, and we think that that's what's driving our superior performance versus other virtual cell approaches now. And so I would say assembling the data and putting in the work to make it useful, that's one part. That one key limitation that is actually, I'm pretty sure even for Lilly, it's been harder to solve that than generate more data. That's clean and machine learning grade. It's harder to say, okay, we have decades of data, how do you actually get it together to train something new? And then another bottleneck, as I mentioned, will really be your utility. We actually don't see, let's say, the model part, the AI architecture part as a key limitation today, at least not in the virtual cell space. I think for us it's how do you get enough data together to train a single model? Maybe that's the architectural thing you have to get right. And beyond that, it's how do you actually integrate this into the workflow of biopharma? Because I think, as David will talk about, when it comes to training on patient data and patient models, there's, I think, very logical ways to try to integrate it into the workflow, and it's less clear in the discovery setting.
Thank you. And maybe a last questions for you, Sabi, but short answer.
This is the last one, yeah, sure.
Short answer.
I'm sorry.
As an insider, what is your opinion on the current trends observed in the use of AI for drug developments?
A short answer. Yeah, I think digital pathology and patient foundation models are super interesting. We have to figure out I think we'll have to see whether the current big wave actually is more successful than the previous one, but again, the other gentleman will talk more about this. I think from our perspective, we see that drug design is something that is a much more quote-unquote solved problem with AI, and their AI has really proven to be a useful tool. And what we care about the most, of course, is how do you actually now take AI to the next frontier between these two worlds and use it to predict the outcome of biological, more complex biology, and how do you use that to actually inform drug discovery to be more rational, and how do you reveal sort of novel insight with that?
Thank you. Go to you, David. First, tell us a few words about Bioptimus in terms of activity, but also strategy.
Thanks a lot, Loïc, and thank you for the invitation. It's a pleasure to be here with you today, with my fellow panelists. So we created Bioptimus 2 years ago with one mission: to develop large-scale, universal foundation model for biology. And think of it, when we speak about digital twin, as the brain behind the digital twin. The ability to encompass various biological modalities to then simulate the biological processes that Thomas was thinking about. And we created this company 2 years ago because my previous team and myself, we've been developing AI solutions for 100 different kind of needs and problems, using every time specific data to solve a specific problem. And you see there is a new paradigm that has came a few years ago now that changed everything. And the new paradigm is that first, we can train foundation model across task, across disease, across situation, across context that can benefit synergistically from all these different inputs. And the second thing is that biology is multimodal. It's not only one modality such as text or transcriptomics or proteomics that will fully describe the underlying biological processes. So you need to go fully multimodal from day one if you want to really solve the problem. So that's why we're after, and that's what we are building.
And can you give us a word about H-Optimus 1 and how you will build from here to come closer to the full digital twin visions?
Yeah, absolutely. So it has been an exciting journey because we released this year our first model called H-Optimus 1. It's a foundation model for digital pathology. So what is digital pathology? You can think about as the science of cells and tissue. And it's considered as the gold standard in many diseases, especially solid tumors in oncology, to be able to diagnose the patient, understand the subtype, and understand which treatment will be the most efficient for a given patient. And it has been an exciting release for the field because today this model is one of the most accurate models in the world. It has been downloaded millions of times. It's used by thousands of users across the world from San Francisco to Singapore. And it has been tested by hundreds of scientific papers on several hundred of tasks. The value of this kind of model is to help the scientists identify better biomarkers or develop better diagnostics tools using a smaller amount of data set, which is exactly what is needed to improve treatment outcome. And we are not the only one in the field. Thomas is, of course, one of the experts also here. But it's very important, I think, to have this kind of innovation come to the researcher and to the patient to help them develop this new biomarker and ultimately better treat patients. What is interesting also is that if you think of this digital twin and our personal vision at the end, which is to build multimodal reasoning model that can reason across scale. So from molecules to cell to tissue, tissue to organs, organs to patient. Histology is a good choice for two reasons. First, it's where AI already works quite well. It's a quite mature task for AI in biology. And secondly, if you think about this journey, it's quite in the middle. And it's a great platform to expand them from the molecular side on one hand, and for the clinical side, the macroscopic side on the other hand. And that's exactly our vision at Bioptimus. Now we are focusing a lot on building other modalities on top of our histology model to not look at the tissue only, but look at what's happening within the tissue at the proteomics level, at the transcriptomic, at the genomic level, and then later on at clinical, patient level data also on top to go closer and closer, ultimately, to this real fully fledged digital twin of a patient.
And from your experience, what do you see as the main challenges for AI in health? Very broader questions and a smaller time to answer.
Okay. First thing is data. I'm going to be quick because I think everybody aligned since 9 a.m. on this. My twist is that we talk about data, but not that all data is the same. Of course, scale and sample size is important, and we need to go order of magnitude beyond what is available today. The second thing is the multimodality. Having millions of insurance claims, for instance, or histology images is not enough to build a real digital twin. You need to have multimodal data, so for the same sample, to have the genomic, transcriptomic, proteomic, ideally, imaging, outcome. And this is a data that is very hard to get and to generate. And this is where we are working with the ecosystem to unlock and generate novel data at scale. Third thing is the representativity of this data. You don't want to have a digital twin of the French biology or the US biology. You want a digital twin for the world, so it means that you need to have access to data across different geographies, across different technologies, and be able to combine that together. That's the first thing, data. The second thing is how do you build the science and the architecture of the model that can leverage this data? And we have to be humble here. It's an open scientific question. There are a lot of different approaches. My colleague is starting from the cell, going to the patient. We are going from the tissue, going to the cell and to the patient. Many valuable approaches are here, and it will be very interesting to see what are the best approaches to tackle this problem. Third thing is the user and the patient and the researcher. Because at the end of the day, we want to impact the patient. A model is not enough. You need to build a bridge between the model and the patient and the researcher. And this bridge can be application, For instance, we're looking at Okin coming after, that is building the environment to leverage this kind of model. It can be chatbot. We are working also with AWS that is working a lot of agent. There are a lot of different ways to leverage this model. And ultimately, it will be a mix of everything that will be needed to bridge the gap with the user. And finally, if you allow me, Loïc, it's about trust. It's about trust and regulation. We need to go step by step to build trust in the model, to benchmark it very closely. There is good initiative looking at the European Society of Medical Oncology, for instance, has published guidance a couple of months ago to explain what do we need to prove to the clinician in order to letting them use our model for a given task. And we are on this journey to bring our model to the patient. It will take several years.
No, I guess regulation is a critical topic to make it happen, especially in the EU. And listening from you, I think what is the most interesting is the different perspective that you have shared, guys, between mid-term statistical model with already powerful progress, and I would say more long-term fundamental research approach to build intelligence model able to deal with hypotheses and expandability. Now I'm going to ask the same question to each of you. In a nutshell, what is the main message you would like to share with the audience as a takeaway? And Thomas, you have more time than the two guys.
Yeah, you overshot, so now you have more time.
Okay. So what's the main message? So at Lilly, running AI at Lilly is allowed us to really have insights to AI throughout the whole value chain of a pharmaceutical company. So that's drug discovery, small molecules, large molecules, genetic medicines. That's then trial design, for example, enrollment, then a lot of language models to, for example, write documentations. The submission of our latest drug was for the FDA was literally 500,000 pages long. So nobody's going to read that, and you need AI to write it, actually. And then manufacturing and commercial and finance and so forth. And one thing which is quite clear in all these areas is to really have impact, especially for the startups out here, is you have to do the hard stuff. So companies like ours, we are going to get pitched by startups who do another wrapper around ChatGPT 5 times a week. And you have to imagine everything you can do in 2 weeks, we can do as well in 2 weeks. So the only thing that actually brings value are the difficult things, to go really deep. And that's why it's a pleasure to be on the stage with colleagues who actually do that in virtual cell and in pathology. And that takes many, many years. That needs very deep expertise in your area of application. Language will not solve that. Large language models will not get us to AGI, and they will not get us to new medicines. You need these dedicated models. You have to invest in compute, in data generation, in understanding these problems. So there's no way around to doing actually the hard stuff. Right? And the beauty about it is that's actually the interesting thing, right? If you think about the reason we have language models these days, that they can write like Goethe or Dumas or somebody else, is that language is structured, it's rather easy, it's produced by our brain, it's not so old, maybe 200,000 years. But the reason there are no robots is running around is that even digit manipulation needed millions of years of evolution. And a single cell we are dealing with, right, has a mechanics of billions of years of evolution. That's a complexity our human language falls short to even describe. And that's why we need dedicated models, dedicated foundation models. And that's why I'm so optimistic and excited about AI and machine learning and all the compute we have now, because we can actually use it for good instead all the LLMs just for social media. So that's, I think, that should be the take-home message. Do the difficult thing, enjoy it, dive deep, and that will actually make progress and at the end of the day help patients, because that's why we are here. We want to build AI to lessen human suffering, and that's the route to do it.
Thank you, Sabi.
That's really hard to follow. But no, it was amazing, and I completely agree. I mean, 2 things I would add is, A, especially in our field, right? Virtual cells, virtual biology, don't try to compete on data. I think in our world, it's becoming more and more commoditized in the sense that you have access to more and more cheap, higher and higher grade and larger data sets that are generated in simple biological models. So instead of saying, okay, I'll generate another large data set and that's what will make me win, instead of that, think about how do you bring together data from multiple companies, multiple sources, and then of course complement it where it needs to be complemented in-house. And more importantly, or once you've done this, think about how is this useful and how do we actually not just hit really interesting benchmarks that ultimately nobody in drug discovery or translational medicine will care about, but how do we build something that's useful for them now, day to day?
Thank you. David?
Thanks. Fully echo what my colleagues are saying. Personally, I'm so very grateful about living in this special moment in time and space, because we'll see in our lifetime, I hope, very drastic changes coming thanks to these technologies. I agree with Sabi's view on data. However, my call to action is still on the data side, because what is true on the perturbation or cell level data is not true on the clinical data. And to share something with you, I think it's a sovereignty question for Europe at this point. You will have digital twin being trained in China, in Asia, in the US, in Europe. I hope that this technology will be trained using all the different universal biological data that can exist. But so far, what we are seeing is that some continents are going much faster than Europe as generating this very large-scale clinical data set. And I would like the next generation of digital twin to also use European data set and be aligned with European biology in a way and a response to treatment. And I'm looking forward as a call to action, working with this ecosystem to accelerate and think bigger about how we could build together.
Thank you. So it is time to wrap up. First, I would like to thank our panelists. I was also thinking this morning how I can do a sort of summary of this session, which was very, very hard. But I would like to leave you with one key food for thought. So first, to answer the original questions. So AI and digital twins are not the hype of the moment. They are fundamentally reshaping how we discover, but also how we test therapies. But unlocking their full potential requires more than data. It demands biologically faithful model, robust across scales, and capable not just for predicting outcome, but also of explaining them. And the next breakthrough will come from integration kinetic cell twins, tissue level dynamics, and agent able to explore reasons. I hope I did not change the message, but those who master this convergence, for sure, will redefine the future of drug development. Thank you for your attention.
Thank you.
Thank you.
Thank you.